Good afternoon, everyone.
May, may I know who
Yes,
is
sir.
speaking?
Yes, sir.
May I know your name, please?
My name is Prakriti Diwan, sir.
Pragati.
Prakriti, sir.
Okay. So, Pragati, you are a faculty there?
Yes, sir.
Okay. Now you can start.
Thank you, sir. Good afternoon, everyone. I, Prakriti Diwan, once again welcome you all for our today's last lecture session. For today's session, we have Dr. Sanjay Jain as our guest speaker. So let's start our session with the permission of Dr. Jain.
Hi, you can start.
Thank you, sir. We are very fortunate to have Dr. Jain as our guest speaker. Dr. Jain is a professor of pharmaceutics and presently head of the Department of Pharmaceutical Science, Dr. Harish Singh Gour, Central University, Sagar, MP. Chairman, Council for Industry Higher Education Collaboration. Chairman of University Innovation Council. Chairman of University IPR Cell. He, he was also former Dean of School of Engineering and Technology. With over 30 years of experience in teaching and research and 2 years in industry, he has more than 190 research publications with more than 50 H index and 199 I-10 with over 12,000 citation index. He has also written one book and more than 25 book chapter in different national and foreign reference book. He has one US and one European patent to his credit and also filed 3 Indian patents. His main research area is design and development of various new drug delivery systems for the treatment of disease associated with gastrointestine and brain. Recently, he has been conferred with India's most prestigious and highest award, Visitor Award 2018, by His Excellence Sri Ramnathji Kovind, Honorable President of India, at Rashtrapati Bhavan, New Delhi on 2/5/2018. Recently, he has been ranked under 2% world scientist by Stanford University, USA. Professor Jain has also been awarded thrice 1992, 2003, and 2006 with the IDMA Award for the Best Research Paper. Besides, he has been conferred with prestigious ML Khurana Award 2003, CRS Pfizer Innovation Award USA 2004, Best Research Award in Pharmaceutical Technology 2005, Award for Research Excellence by European Institute of Scientific Research, Netherlands, National Teacher Excellence Award 2012, and Lifetime Achievement Award 2019 for his excellence in teaching and research. He has successfully guided 26 PhD and more than 150 M.Pharm dissertations, as well as has completed 8 major research projects funded by various national agencies. Dr. Jain has presented number of research papers in various national and international conferences and delivered many invited lectures in various conferences on different topics. He is member of editorial board of critical reviews in therapeutic drug carrier system, Bagel House UK, regional editorial of current signal transduction therapy, Bentham Science Publisher, and Bentham Science and brand ambassador. He is also referee of various national and international journal of repute. He is also chairman of University Technical Committee and member of board of studies of various universities. He is a life member of many learned societies and has organized many training programs and conferences. May I request sir to join with his deliberation?
Sure. Okay. Thank you, Prakati, for giving my introduction to the participants. First of all, before starting my lecture, I would like to thank organizer of this short-term training program, which is AICTE sponsored, and inviting me to deliver a lecture on the theme future trends in research methodology for drug discovery and development. And under this theme, I would like to tell something about the colon-specific drug targeting. Why I have selected this colon-specific drug delivery, because delivering drug to the colon is very difficult, and for the treatment of the diseases associated with the colon is really very difficult, because it is not easy to deliver effective concentration of drug to the colon. However, there are many formulations are available in the market, but their efficacy is very less, or you will require to deliver high dose of the drug to deliver effective concentration of the drug to the colon for the treatment of the disease associated with the colon. So I thought that this topic will be more relevant to the theme of the this short-term training program, and we'll discuss the challenges and opportunity of colon drug targeting, and we'll see that how you can develop a colon specific drug ladies, how easily you can develop. Right after my lecture, you will come to know that developing a colon targeted drug delivery system is really not a difficult task. It's an easy task. Only is you must have some knowledge, basic knowledge of this drug delivery system. So, well, this my slide is visible now. Anyone can tell, yeah, if it is visible. If you have any problem during my presentation, you just unmute your mic and you can tell me.
Yes, sir.
Otherwise, it should not be one way. I'm delivering a lecture. I don't know whether you are receiving it properly or not. So if you have any at your end, if you have any problem, you please let me know. We'll try to solve that problem.
Sure, sir.
Right. So this my slide is visible now.
Yes, sir. First slide
So
is
as
visible.
as I told you that I shall be delivering a lecture on colon targeting drug delivery, challenge and opportunity. And generally, in general, what type of challenges pharmacists or scientists who are working on the development of drug delivery system are facing? Uh, the general challenging the pharmacists or scientists are facing, that prevention of drug from the biological degradation. This is a major challenge pharmaceutical scientists facing in the development of this drug delivery system. You must have to prevent first. You must have to see whether the drug which you are delivering to the particular organ for the treatment of particular disease, you must have to see what will be the fate of that drug in the biological system after its administration. Whatever route of administration you select, whether it is oral route or parenteral route or any route, you are delivering the drug, but you must have, after reaching the drug in the biological system, what will be its fate? Whether it will absorb or not. Uh, if it is absorbed, how it will be distributed. If it is distributed, how much concentration of drug will be distributed to a particular organ, that you must have to know. What will be its metabolism? What will be its degradation in the biological system? Because drug will be the foreign molecule to the body, and so many system of your body protection system will be acting on the drug to just eliminate the drug from the biological system. So you must have to know the fate of the drug in the biological system after its administration. This is very important. You must have to know during the selection of the drug for particular drug delivery system, and that we are designing for treatment of particular disease. Then patient compliance. Yes, to whom you are delivering this drug delivery system? To whom? The patient. Our first user of our dosage form or drug delivery system is the patient. So whatever drug delivery system we design, we must have to concentrate to the comfort of the patient. The patient should feel comfortable, right? And so patient compliance is very important. Then cost effective. Naturally, country like India, we are facing, we are not, we can say that we are not so rich to afford the treatment of any disease. If any one person of your family is facing any disease, and treatment is nowadays is very costly. So it is a need of the country to develop such delivery system which are cost effective. So we must have to keep during the development of drug delivery system, during the development of any dosage form, we must have to think about the cost effectiveness. The product life extension. This is also related with the cost of the product. Product life extension. If the shelf life of the product which you have developed is less, then naturally its costs will be very high. So if you are if you are able to extend the product life, or shelf life of the product which you have developed, then naturally for more time period you can keep that drug. It will not be spoiled, and it will be cost effective. It will also affect the cost of the product. Then maximize drug utilization. This is related with the targeting. Maximize drug utilization. Whatever drug you are delivering to the biological system or to the human being, then whole drug, say 100 milligram is delivered to the body through any dosage form or drug delivery system. If 100 milligram completely utilized, then that will be the best drug delivery system. So we must have whatever drug you are delivering to the body, it should maximally utilize by the body. If it is not utilized, means 50% drug is degraded, 25% drug is. Hello. I think everything is okay. Hello.
Yes, sir.
Uh, Everything is okay. I receive a call now. I don't know whether it is from your side or somewhere else. Telephone. Anyway,
No, sir.
so, okay. Uh, so we must have to think about its maximum utilization after its administration. If it is completely utilized, means if the drug is after the administration, if the drug is 50% is unabsorbed, means 50% of administered drug is wastage from the body because it is not absorbed. It is not reaching to systemic circulation, right? It is not reaching to the particular end. Say very less amount of the drug will be reaching to the target organ where the delivery of the drug is required, where the treatment of that disease is required for delivering to the particular organ. Then we must have to think about it. Maximum means whatever drug you are administered, if it is completely utilized, that will be the best drug delivery system. And that can be achieved by effective targeting. Effective targeting means the drug which is you are delivering, right? Whole concentration of drug will reach to the target organ, the disease organ, where the treatment is required, right? If you are able to develop a drug delivery system with effective targeting, then the dose and cost of that product will be reduced. This is about when you are at the floor of the drug development or drug discovery for the treatment of particular disease. You must have to know about the drug dynamics in the body. Drug dynamics means the movement of the drug in the body. That you must have to know. After the administration of drug, how it will be moving into the body, because this will affect this by the movement of the drug in the body. You will get a beneficial or non-beneficial effect of the drug. So you must have to know about the movement of the drug. You must have to know the dynamics of the drug in the body. So this graph you might have seen in various uh, textbook of the biopharmaceutics, the onset of action after the administration of the drug, when you will get the onset of action, when the drug concentration will exceed the minimum effective concentration, you will get the response of the drug. If it is a drug concentration in the body is exceeding the minimum effective concentration. But apart from that, if the concentration is exceeding the maximum safe level, you will get the some toxic effect of the drug, side effect of the drug, along with the beneficial effect. You will get the toxic effect or side effect of the drug. So your intention, being the pharmacist, your intention is to keep drug concentration in this therapeutic window. Therapeutic window means the minimum drug concentration should be above the minimum effective concentration and lower than the maximum safe concentration. This is the therapeutic window. So your intention is to keep drug concentration in the therapeutic window for a longer period of time. If you are able to develop such delivery system by which you can easily achieve this minimum effective concentration, means short onset of action, and you are able to keep the concentration in this therapeutic window for a longer period of time, means duration of action of that drug will be longer. That drug delivery system will be the best drug delivery system. So your duty, being the pharmaceutical scientist at the research and development floor, your duty is to keep to develop such delivery system which will keep drug concentration in this therapeutic window. Then since we are talking about the drug targeting, we must have to know a little bit about the drug targeting. Drug targeting is selective effective localization of drug to the vicinity of the target organ. Target organ means if you are intend to deliver to the liver, it should go only liver. It should not go to other part of the body. That will be the effective targeting. It's a selective effective localization of the drug to the just near to the target organ, right? So that you can maximize drug utilization. You can reduce the drug side effects. So why drug targeting is required, or why targeted drug delivery is required? Because for exclusive delivery to the specific pre-identified organ of the body, right? If you it is required, so drug will, if it is a target specific, drug will deliver to the pre-identified organ only, right? It will not go to the other unwanted organs. It reduces the drug access by other relevant non-target organs, right? Drug should not go to the other organs where the action is not desired. Reduces drug toxicity. So this approach will reduce the drug toxicity and side effects, maximize intrinsic activity of the drug, because maximum amount of the drug will reach to the target organ or identify organ, pre-identify organs. So it will maximize intrinsic activity of the drug. Then there are different level of drug targeting. In general, there are two types of the targeting: active targeting or passive targeting. Active targeting that facilitates the binding of drug carrier to the target cell through the use of the ligand. There are some receptor over the cell, and that receptor can be used for delivering the drug, right? That will be the active target. Receptor, that is also known receptor mediated localization of the drug, or receptor mediated localization of the drug carrier. So there are again three type of the active drug targeting: first order, second order, third order drug targeting. First order means the delivering drug to the whole organ, say liver, or second order means to cellular liver cells. Second order targeting is to particular liver cells, targeting drug to the particular liver cells. And third order targeting, that is the most specific targeting, that is the intracellular targeting, means inside that cell you are targeting the nucleus of that drug, you are targeting in the cell. So that is the third order targeting. So this is about the active targeting. Now passive targeting. Passive targeting is body natural response of the drug is used to deliver the drug, for the uptake of the drug. Say RES, reticular endothelial system uptake of the carrier system. That RES system is present mostly in the liver and spleen. So by natural process, by the natural responses, drug will be received. Well, drug will be uptaken, right? So that is the, if you want to target liver or spleen, you can use RAS uptake of the drug carrier system. Now come to the colon. Since colon, as you know, it is the last part of the drug, and delivering drug to the colon through the oral route, it is not easy task because drug has to face so many hurdles, so many barriers, because intestine is about it's a very long intestine, about more than 1 meter in length. So that there are so many enzymes, so many variation in the pH, right? So that drug may get affected or get before reaching there, drug may get absorbed from the upper part of the GIT, right? So we must, if you are targeting drug to the colon, you must have to know anatomy, physiology of the colon. Only then you can target drug by designing suitable drug delivery system. The colon forms the lower part of the GIT. It is the last part before the rectum. It is the last part of the gastrointestinal tract, extend from ileocecal junction to the anus. That is the colon. And colon is upper 5 feet of the large intestine, right? And rectum is lower 6 inches. And if you see the anatomy of the colon, the colonic membrane will be having so many membranes. The outermost, that is towards the mucosal side, the mucous membrane, then below that, submucosal level. Below submucosal level, you will find a circular mucosal layer, and below circular muscular layer, you will find longitudinal muscular layer and serosal. So it is a very complex membrane. Colonic membrane is very complex membrane. It is very difficult to cross this membrane. If you want to absorb the drug which is delivered to the colon through the colonic membrane, it is not easy task. There, apart from the anatomy, physiology, apart from the structure of this colonic membrane, there are so many barriers in the colonic membrane that will prevent the absorption of the drug from the colonic mucosa or colonic membrane. Now, the colon, as you know, the colon is the last part of the gastrointestinal tract, and it is a hostile environment. Long colonic transit time. If you in coming slide, you will come to know the transit time of the different part of the GIT. Then you will find that colon or large intestine is having the largest transit time. Means the residence time of the drug into the colon is 20 to 30 hours. Means if we have 20 to 30 hours, drug is lying in a particular organ, you can play with that organ. You can play with that drug, right? And then it is having unique microbial flora. In the coming slide, you will come to know that colon is full of microorganisms, aerobic and non-aerobic microorganisms. It is full of microorganisms, and that microbial flora can be used for releasing the drug from the developed drug delivery system. It is highly responsive to the absorption enhancer. The colonic membrane, which we have seen in the earlier slide, the colonic membrane is highly effective for the absorption enhancers because it is colonic membrane colonic membrane is having the colonocyte tight junction, occlusive tight junctions. So through this paracellular transfer of the drug molecule is not possible from the colonic membrane. You will require, if you want to get absorbed from the drug from the colonic mucosal side to the serosal side, to the blood circulation, right? You will require to change the permeability of the colonic membrane by using some absorption enhancers. And it is the colonic membrane highly effective to the absorption enhancers. There are two routes by which you can achieve this colonic delivery, or by oral administration or by rectal administration. No doubt, by rectal administration, drug has to travel very low distance, very smallest distance. But if you are delivering drug through the this oral route, then it has to travel long distance, right? But administration of the drug through the rectal administration is not convenient. It's difficult. It requires some experts for delivering drug to the colon through the rectal route. So that rectal route is not suitable for colon delivery of the drug. So we were talking about the transit time. Here you will see that length and pH and transit time of different parts of the GIT. This is the GIT, okay? And starting, this is the stomach. Stomach is the upper part of the GIT membrane after the esophagus. It is having length 20 centimeter and pH is ranging 1.5 to 2. And the residence time is 1 to 3 hours, right? Or we can say the transit time is 1 to 3 hours. And after that, the small intestine will come. Although this is not a small intestine, but it is lengthwise still very lengthy as compared to other part of the GIT. The upper part of the small intestine is duodenum, which is having pH 4.5 to 5. Then jejunum, which is having pH 5 to 6.5, and last part is of the small intestine is ileum, which is having pH 6 to 7.5. And this total length of the small intestine is approximately 730 centimeter, and the transit time is 3 to 5 hours. If a molecule that to be delivered through the oral route and it has to cross this small intestine, it will take 3 to 5 hours to come out from the duodenum to ileum part, last part of the ileum. Then after the ileum, then you will have this ascending colon. This is the colon, right? This is the colon, and ascending colon is having the this length 20 centimeter and pH 6.4 plus minus 0.6. Right? If you see the pH of the last part of the small intestine is about 7.5. Then sudden when the content will come into the ascending colon, which is very adjacent to the ileum, right? Or when it crosses, it reaches to ascending colon, there will be sudden fall in the pH. Then sudden fall in the pH of the content of the ascending colon is due to the presence of short chain fatty acid, which is arising from the bacterial fermentation of polysaccharides. The polysaccharides present in the content, which will be degraded, and you will get the short chain fatty acid. Like lactose is fermented to the lactic acid. In formation of lactic acid, there is sudden fall in the pH. So the pH is changed from 7.4 to 6.4, sudden fall in the pH. Then the transverse colon. Transverse colon is having length 45 centimeter, pH 6.6 plus minus 0.8. Then descending colon will come. Descending colon is again having 30 centimeter length, and pH is gradually rising from transverse colon to descending colon. Now it becomes 7 plus minus 0.7. And last part is sigmoid colon, having length 40 centimeter, and pH is 7 to 8. And the total transit time is ranging about 20 to 25 hours, or sometimes you will get 48 hours. Means if the content will remain in the colon up to the 48 hours. We were talking about the microflora in different regions of the GIT. Say there are in the microflora that is present in the upper part of the GIT. When I say upper part of the GIT, it means the stomach and small intestine, right? In the upper part of the GIT, as well as the in the ileocecal region, there are only 6 microorganisms present. But if you see the microorganisms, list of the microorganisms that are present in the colon, in this colon, there are a long list of the microorganisms, long list of the aerobic and anaerobic microorganisms, say bacteroids, bifidobacteria, eubacterium, Peptococcus. They are a long list. And if you see the 30% of dry weight of the feces consisting of bacteria. Means if you see the bacterial count, if you count the bacteria in the feces, then you will find approximately 30% of its weight consisted of the bacteria, right? That are coming from the large intestine or colon. And these bacteria, if you see the bacterial count in different part of the GIT, then you see in the stomach is having very less amount of the bacterial content, 0 to 10 to the power 3 colony forming unit per ml. Jejunum, that is the middle part of the small intestine, that is having the bacterial count 0 to 10 to the power 5 micro colony forming units per ml. And ileum is having little bit higher amount of the bacterial count, that is a 10 to the power 3 to 10 to the power 7 colony forming unit per ml. While if you see in the colon, 10 to the power 11 to 10 to the power 12 colony forming unit per ml bacterial count you will observe in the colon. So these these bacteria can be used. They are generally beneficial bacteria. They are releasing number of enzymes, so number of reactions are taking place, and these bacteria excretion can be used for the drug release from the developed drug delivery system. So these colonic bacteria releases 2 types of the enzymes, reducing enzymes and hydrolytic enzymes. Under the reducing enzymes, the colonic bacteria are releasing, that is a nitroreductase, azoreductase, N-oxidase reductase, and sulfoxide reductase. Under the hydrolytic enzymes, they releases esterases, amidases, glycosidases, glucuronidase, and sulfatase. These are the hydrolytic enzymes. And these this reducing enzyme and hydrolytic enzymes, these enzymes carrying a number of metabolic reactions in the colonic regions. What are those metabolic reactions? The hydrolysis, reductions, decarboxylation, dealkylation, dehalogenation, deamination, acetylation, and esterifications. Under this azoreductase, generally for the development of colon-specific drug delivery system, azoreductase enzyme, which is released by the azobacters present in the colonic lumen, and this azoreductase enzyme is used for releasing the drug from generally from the prodrug, like which are having the azo bond, and this azoreductase enzyme is responsible for degrading, for cleaving the azo bond. So this azoreductase enzyme can be used for triggering the drug release. Polysaccharidase, no doubt, of course. We can use polysaccharidase enzyme also, which degrades the polysaccharides. So many gums, the gums we can use for delivering drug to the colon. And polysaccharidase enzyme will be acting, and that polysaccharidase enzyme will digest the gum and drug will be released. So as I told you, there are so many barriers are present in the colonic lumen, and that produce hindrance for the absorption of the drug, for the transfer of the drug from the colonic mucosal site to the serosal site to the blood circulation. These are the barriers. Small surface. It is, if you compare the surface area of colonic um, colon, then it is much less than the small intestine. So it has a small surface area. So it will have limited absorption of the drug. And there will be specific and non-specific drug binding with the dietary component and product released from the colonic bacteria. So they may form specific and non-specific drug binding, and if it is bound with the dietary components or the product released from the colonic bacteria, then the drug will not absorb. In the colon, as you know, that only water is absorbed from the colon, and some minerals are also absorbed. So it is having the colonic content is having high viscosity, which impedes the diffusion of drug from the lumen to the mucosal site. So this is the physical barrier you will find in the colonic lumen. Mucus layer. If you compare the thickness of the mucus layer, which is present at the surface, epithelial surface of the colon, in the colon you will find very thick mucus layer, which retards drug release reaching to the epithelial surface for the absorption. And the colonic membrane, you will find one more barrier, that is unsterile water layer, which is present just below the lipophilic membrane, epithelial membrane, or mucosal membrane. You will find unsterile water layer, which is a barrier for the lipophilic drug. It is hydrophilic one. And the epithelial membrane of colon, as I told you, it is having colonocyte or occluding junctional complex in the colonic epithelial layer, which provide physical barriers. So because of presence of cells, there is no intracellular space present. So through that, paracellular transfer of the drug is not possible through the colon. Epithelial surface, it is highly lipophilic layer, lipid bilayer, so it produces a barrier for the hydrophilic drug. As I told you, because of these barriers, the absorption of the drug from the colon is very low. Only water or dissolved minerals are absorbed. This is a pictorial depiction of the conventional and colon-specific dosage form. If the drug is administered in the conventional dosage form through the oral route, right? Say in case of the, we take a simple example of the tablet, oral tablet. We'll take it through the oral route. It will go into the stomach. Tablet will disintegrate, and the particles, small particles, drug will start dissolving in the stomach fluid, gastric fluid. Or if it is not absorbed, then it will reach into the small intestine, where most of the orally delivered drug will get absorbed, right? A very small amount, very small amount, unabsorbed amount will reach to the colon. So if you want to treat the diseases associated with the colon, then you will require to deliver more amount of the drug. Only then effective concentration of drug will reach to the colon. Otherwise, what will happen? Drug will get absorbed from the upper part of the GIT, and from the circulation, it will reach into the colon. In the next day, I will explain how it will reach to the colon if the drug is delivered through the conventional doses. But if the drug is delivered through the colon targeted drug delivery system, then this green one, drug will remain intact, or dosage form will remain intact in the stomach, and it will, after adequate residence transit, it will go into the small intestine. It will be intake in the small intestine, this green one, And when it will reach to the colon, the drug delivery system will be uh, cleave or degrade or break. Then you will get the drug, free drug in the colon. So sufficient amount of the drug you can deliver to the colon by colon specific drug delivery system, and a small amount of the drug will require to deliver if it is delivered through the colon specific target colon specific drug delivery system. Say this is systematic circulation. This is about the conventional dosage form. As I told you, if you want to deliver a drug to this colon for the treatment of colon-specific drug delivery system, you will require to deliver high amount of the drug. This thick layer, high amount of the drug you will require to deliver. So most of the drug will absorb, which is delivered through the conventional dosage form. It will get absorbed, it will reach to the blood circulation, and through blood circulation, it will come back to the colon, right? But if the drug is entrapped in the colon targeted drug delivery system, then what will happen? Very little amount of the drug will get absorbed from the upper part of the GIT. The whole amount, the most of the amount that is present in the colon specific drug delivery system will be delivered to the colon. Say there is one example that you can easily correlate it with the this difference. You can easily understand the difference between the colon specific drug delivery system and conventional dosage form. Say metronidazole. आपने metronidazole देखा होगा. Metronidazole tablet कितना लेते हैं? 500 milligram 3 or 4 times per day, means 1.5 gram to 2 gram, just to kill a little teeny amoeba that is present in the colon. उसको मारने के लिए आपको 1.5 gram to 2 gram metronidazole लग रहा है, right? So what क्यों इतना amount, why you are delivering such a high amount? Because for killing tiny amoeba, you will require very micro concentration of the metronidazole, right? So what will happen if you deliver metronidazole in 500 milligram 3 or 4 times a day, then it will get absorbed. It is having a tendency to absorb from the upper part of the GIT, right? It will get absorbed, it will reach to systemic circulation, and through systemic circulation, very little amount of the metronidazole will come to the colon, and then that metronidazole will kill the amoeba, right? This is in case of the conventional dosage form. If you make colon specific drug delivery system of the metronidazole, then very little micro concentration of metronidazole will require, because whole concentration you are delivering to the colon. It is not releasing drug in the upper part of the GIT. So in that way, you can maximize drug utilization, you can reduce side effect of that drug, because drug metronidazole is reaching to the blood circulation. आपको nausea, vomiting feel होता रहेगा all the time. But if it is delivered through the colon specific drug delivery system, you will never feel nausea, vomiting, because drug is not reaching to the blood circulation. It is locally acting and reaching to the colon, delivering drug directly to the colon, means the site of infection where the action is desired. Now, which candidates you can use, which drug candidates which you can use for designing the colon-specific drug delivery system? Those drugs which are having poor viability from the stomach or small intestine, like peptidal drug, or those drugs which are having deleterious effect on the gastric mucosa, like non-steroidal anti-inflammatory drugs, so that affect the mucosal membrane of the upper part of the GIT, that can also be designed in the colon delivery system, or drugs required for treatment of the diseases associated with the colon, like amniosis, colon cancer, ulcerative colitis, inflammatory bowel disease. In all such cases, if you are able to design, you are able to deliver drug to the colon, then it will be most effective drug delivery system, especially for the treatment of the disease associated with the colon. These are some examples of the drug candidates for colon-specific drug delivery system. Digestive enzyme supplements for the treatment of the cystic fibrosis, pancreatectomy, chronic pancreatitis for local action, and sulfasalazine, ipsalazine, these are our valsalazine, mesalazine. These are the product of the prodrug of the 5-amino salicylic acid. These are used for the treatment of inflammatory bowel disease, Crohn's disease, or irritable bowel syndrome for topical use. Prednisolone, even prednisolone can also be used for the treatment of these colon-associated disease for topical actions. 5-fluoro uses for colorectal cancer, for systematic absorption, non-steroidal anti-inflammatory drugs, prevention of gastric irritations, thyroid, vaccine, peptidal drug, for oral delivery of vaccines, insulin, for oral delivery of peptides. So for systematic absorption, these drugs can be used, can be delivered through the colon-specific delivery system. So oral colon-targeted drug delivery systems, those releasing exclusively in the colon, those systems releasing drug exclusively to the colon, they are called colon targeted drug delivery system, and it is increasing colonic viability. If you are able to exclusively deliver drug to the colon, it will increase colonic viability of the drug, enhance therapeutic efficacy by localizing to the colon. Naturally, if the more amount of the drug is present at the desired site, the therapeutic efficacy of the drug will increase, reducing systematic adverse effect by reducing absorption of the drug from upper intestine. Right? Drug directly reaching to the colon. It is not reaching through the systemic circulation, through the blood, right? After the absorption. So drug directly reaching to the colon. So it will reduce the systematic adverse effect of the drug. Maximize drug utilization because it is a target specific, it is organ specific, it's a colon specific. So it will maximize drug utilization. Reducing doses of the drug naturally, as I told you, for killing a tiny amoeba, you will require micro concentration of the metronidazole naturally. So from milligram to 500 milligram to microgram concentration will reduce. That will also reduce the cost of the product. So reducing the doses of the drug, frequency of the dosing can also be reduced. Make the drug retention time in the colon, because the transit time of the colon is very high. So the retention of the drug in the colon will be more. So it will be drug can be utilized completely in the colon for its effect. Now come to the various approaches for colon targeting. There are different approaches. I have summarized some approaches. Product approach like Azova product, Glycoside product, Cyclodexin product, Dextron product. Site-dependent drug delivery system can also be designed for increasing the colonic viability. Surface charge dependent drug delivery system, just for making the system which will adhere to the colonic membrane. So the drug target time or drug resistance time can also be increased. And then pH dependent drug delivery system like it can be used because there are a lot of variation in the pH from stomach to the large intestine or colon. There is a lot of variation in the pH, and that variation in the pH can be used for designing the drug delivery system for delivering drug to the colon. Biodegradable drug delivery system like pectin or guaiacum, because for degradation of the pectin in guaiacum, specific enzyme is present, polysaccharidase enzyme is present, that will be that is released by the colonic microwave flora, and that will be responsible for degrading the pectin and guaiacum. So we can use some, we can design some biodegradable system using some gums. Then redox drug delivery system, especially in case of the inflammatory bowel disease. Inflammatory bowel disease, the oxygen level, reactive oxygen species (ROS) level is increased, and that increased ROS level can be used for degrading the drug release uh, from the this drug delivery system if it is having some redox sensitive polymer. Then active targeting dependent drug delivery system. Yes, this is the approach. This is very new approach, and on this approach, I receive a President Award. Right, active targeting dependent drug delivery system means there will be a dual targeting. We design drug delivery system which will target drug colon, and from the that carrier will be released in the colon, and carrier will be further targeted to the colorectal cancer. That was designed for delivering drug concentration, especially for colorectal cancer. So the active targeting dependent drug delivery system can also be designed. Time release system, pulse in cap. The first time release system was designed for colonic delivery of the drug. That was the pulse in cap. And time release means you have to delay the release. Say transit time of the stomach is say 2 hours, and transit time of small intestine is say 5 hours. So total say 7 hours. If you are able to delay the drug release from the designed drug delivery system up to the 7 hours, by the time your delivery system after its oral administration will reach to the colon. So by making the time release system, you can deliver effective concentration of drug to the colon. So this is about the Azova drug delivery system. When time will be over, just give me a ring, okay? Or just tell me, okay? So we can just when time will over, you just give me 5 minutes. I will conclude my speech. So Azova prodrug, this is a 5-amino salicylic acid used for the treatment of inflammatory bowel disease. It's a very popular prodrug used for the treatment of inflammatory bowel disease. And the sulfasalazine, the product is sulfasalazine of 5-amino salicylic acid. So when sulfasalazine, which is having this azo bond, when this sulfasalazine is delivered through the oral route, it will be unabsorbed in the upper part of the GIT. It is not absorbing until unless this 5-amino salicylic acid is not free from this product. So, this sulfasalazine will reach, it will be unabsorbed in the upper part of the GIT. When it will reach into the colon, azoreductase enzyme will be acting, which is released, azoreductase enzyme, which is released by the colonic bacteria. It will act on this azo bond. Azo bond will cleave. You will get one molecule of 5-amino salicylic acid and one molecule of sulfapyridine. And the most of the side effect of sulfasalazine is because of this sulfapyridine, which is cleaved from this prodrug, right? So because of this toxicity, some other product of 5-amino salicylic acid were designed. This is the sulfasalazine. By replacing the sulfapyridine molecule is replaced by the 4-amino-benzyl beta-alanine amino acid and 4-amino benzyl hyporate. This amino acid is attached with this 5-amino salicylic acid. If 4-amino benzyl beta-alanine is attached with 5-amino salicylic acid, the product is known as valsalazine. And if 4-amino benzyl hyporate is attached with the azo bond by this 5-amino salicylic acid, the product is known as epsalagine. So this prodrug, this valsalazine and epsalagine, which is having the azo bond. When this drug will reach into the colon, azoreductase enzyme will be acting and cleave this azo bond and double bond end, and you will get one molecule of 5-amino salicylic acid. Then further, 5-amino salicylic acid product was designed, where scientists have used two molecule of the 5-amino salicylic acid, and they join 5-amino salicylic acid with the azo bond. They conjugate it with this azo bond, right? So, and this product is known as olselagine. Olselagine is the dimer having two molecules of 5-amino salicylic acid. When this olselagine molecule reach into the colon, the azoreductase enzyme will cleave this azo bond, and you will get two molecules of 5-amino salicylic acid. This is about the prodrug of 5-amino salicylic acid. Now, prodrug of glycoside prodrug. As you know that I told you about that bacteria present in the colon are releasing polysaccharide, different polysaccharide enzyme. And these colonic microflora are releasing beta D galactosidase, beta D glucosidase, alpha L aravinofurantosidase, beta D xylopyrenosidase. These polysaccharide enzymes are released by the colonic microflora. This is the prodrug of dexamethasone, which is having the glycoside bond. So dexamethasone 21B beta D glucoside, and this product of dexamethasone or prednisone have been prepared with beta glucoside, and beta galactoside. And these enzymes, polysaccharide enzymes, are acting on this bond, that is the glucoside or galactoside, and we can get a product of dexamethasone. Next one is the cyclodexin product. As you know, the cyclodexin, beta cyclodexin is very widely used for enhancing the solubility of the water insoluble drug. And the solubility of the water insoluble drug is enhanced when drug will be encased in this molecule of the beta cyclodexin. When drug is encased, right? So inclusion complex is formed. But if the drug is conjugated with the OH bond group present over the surface of the beta cyclodexin by the covalent bonds, then it will be colon. This beta cyclodexin will become a colon specific. So OH group of the beta cyclodexin is substituted with the diphenyl. This is the example of the diphenylyl acetic acid beta cyclodexin prodrug. So OH group of the beta cyclodexin substituted with a diphenylyl acetic acid through an ester or amide linkage will get this prodrug. It will be ineffective, unabsorbed in the upper part of the GIT. When it will reach into the colon, the polysaccharide enzyme cleave this bond, will get the parent active drug molecule, and beta cyclodexin, which is released from this product, will be excreted unchanged with the feces. And then Dextron product, this is another product where the drug is conjugated with the Dextron. So drugs which are conjugated with the Dextron are exclusively degraded by the dextranase enzyme, right? It is excreted by this dextranase enzyme, and drug is bound with the dextran by this succinate bond or glutarate bond, right? If the dexamethasone and glutarate dextran, if you see this ester of the product, it's having the half-life of 103 hours, and methylprednisolone succinate dextran ester having the half-life 82 hours. Next one is the approach is size-dependent nano-delivery system that improve colonic residence time because of size, nano size. They will adhere with the membrane of the colonic membrane, and their residence time will increase. exerting an epithelial enhanced permeability and retention effect, low preferential uptake of nano-sized particle by immune cell in case of the inflamed regions, especially in case of the inflamed bowel disease. Avoid rapid carrier elimination by the diarrhea. And in case of the inflamed bowel disease, there are chances of the diarrhea. So if delivery system present in the nano size, then clearance from the colon can be avoided. M cell involved in predominant uptake of the nanoparticles through the transcytosis. So their effectiveness of the drug molecule, if they are entrapped or if they are encapsulated in the nano delivery system, can be enhanced. Then surface charge dependent drug delivery system, both type of the charges, cationic or anionic nanocarriers, both are beneficial for the colonic delivery of the system. Say if the nanocarrier is positively charged, it will enhance adherence to the inflamed mucosa of the colon, especially in case of the inflamed bowel disease. It reduces the nanocarrier clearance, which when the intestine motility is enhanced. Mucus layer thickness in the Crohn disease, promising strategy for targeting retention of the drug delivery. So these are the example of the cationic nanocarrier system, colon rate loaded nanoparticles comprising of cationic polymethacrylic means hydrogen RS, chitosan functionalized polylactic glycolic acid nanoparticles, chitosan and pectin coated liposomes. These nanocarrier systems are used for delivering drug to the colon. In case of the negative charge nanocarriers, enhance adherence with the higher concentration of the positive charge proteins. In case of the inflammable bowel disease, when the colonic membrane is inflamed, this eosinophil cationic protein or transferrin are overexpressed over the surface of the inflamed epithelial layer of the colon. So the negatively charged nanocarriers are adhere with this protein, right? So their residence time can be increased and can be make more effective. If you compare the adherence preference with positive, negative, and neutral nanoparticles, then you will find that negative charge nanocarriers are more effective as compared to the positive one and neutral nanoparticles. This is an example of the negatively charged nanocarriers, anionic nanostructured lipid carrier bearing budesonide, okay? And then it's a very popular pH-dependent drug delivery system that you can design in your laboratory also. Just coating with the pH-sensitive polymer. You select those type of the polymers which dissolve at the pH of 6.5 or above, right? So the pH of the 6.5 pH you will find in the region of ileum or ileocecal region. So by the time this coating of the pH-sensitive polymer is dissolved, your system will enter into the colon. So we can deliver effective concentration of the drug by coating the pH-dependent polymer, pH-sensitive polymers. It is because, as you know, that variation in the pH in different part of the GIT, pH of the terminal ileum or colon is generally higher, and coating with pH polymer, or we can design some nano delivery system of pH-sensitive polymer like Udrazit, L and S. You can design some polymer nano delivery system that will be the pH-sensitive. It will dissolve. These nano delivery system will start dissolving when they will get a suitable pH. At a pH of the ileum, say 6.5 or above. So when it will start dissolving, by that time your delivery system will reach into the colon. So we can use different Udrazit, which are pH-sensitive polymers, and they are pH-sensitive. Udrazits are also exhibiting mucoadhesivity, so say having the dual advantage. So this is Udrazit coated nanoparticles, Udrazit coated liposomes we can develop. Intra-individual variability in pH is more if we select pH-sensitive polymer. So for that, some advanced approaches we can use by using the instead of using the single approach, we can use dual approach. From using the pH sensitive polymer, we can use some polysaccharides or biodegradable drug delivery system. So next one is the biodrug delivery system. Next approach, that is, as you know, that a number of specific enzymes are released by the colonic bacteria. There's azoreductase or polysaccharidase, all right, and that can be used for releasing the drug from the drug delivery system, or that can be used for degrading the drug delivery system and releasing the drug. So by using that type of the polymer, or the synthetic polymer or natural polymer, we can use these polymer for either for coating the drug delivery system, or by we can design some nano drug delivery system, right? So we can use azoaromatic polymers. This is synthetic one. We can use alginate, pectin, or Dextron as a natural polymer. These are the biodegradable polymer. They degrades only in the colon. Means these polymer are digested by the colonic enzymes, right? And only in the colonic enzymes. And so we can use these polymers. These are some example. Saffron and co-workers prepared azoaromatic polymer using styrene hammer with divinyl azobenzene as a cross-linker. Means in the structure of the polymer, they have put azo bonds, right? Number of azo bonds. So that say this is the structure of styrene 2-hydroxyethyl methacrylic copolymer, which is polymerized by using the divinyl azobenzene. So in the structure of this copolymer, they will have number of N double bond N, right, azo bond. So when this copolymer, when it is delivered through the oral route, it will be ineffective. It will not dissolve in the upper part of the GIT, but when it will reach into the colon, the azoreductase enzyme will be acting, and all the azo bonds present in the structure of skeleton of the copolymer will be degraded. So you will the polymer become porous one, and drug will be released. So two type of the cross-linker, divinyl azobenzene we can use as a cross-linking agent, or we can use NN bis beta styryl sulfonyl 4,4-diamino azobenzene. That these are two cross-linker we can use for synthesizing the copolymer, azoaromatic copolymer. So as I told you, can either you can prepare some nanoparticles of azoaromatic polymers, right? And these azoaromatic polymers filled in the enteric-coated capsule. In the capsule, and capsule is enteric-coated. And this enteric-coated capsule will be ineffective in the stomach. The enteric-coating will dissolve in the small intestine. And by the time the enteric-coating dissolve, capsule will reach into the colon. The capsule will dissolve. The nanoparticle of azoaromatic polymer will be released, and azoreductase enzyme will be acting. Drug will be. हेलो हाँ ठीक है 12 बजे ना ठीक ओके right. So the drug will be released in the colon. As we can use azoaromatic polymer for the coating purpose. Prepare granules of the drug coated with the azoaromatic polymer, and these azoaromatic coated microspheres will be intact in the stomach and small intestine, because this azoaromatic polymer will not dissolve at the pH of the this stomach or intestine. But when this delivery system reach into the colon, the azoaromatic polymer, the azoreductase monomer will reduce the azo bond present in the aromatic polymer coating become porous one, and drug will be released. Or matrix type system, we can prepare a drug and polysaccharide means square gum. We prepare a matrix, mix and compress it. We can prepare a compressed matrix tablet, and that will be intact. When it will come in contact with the fluid of the stomach and intestine, there will be little bit swelling of the uh, this matrix tablet, or some of very little amount of the drug will release because of swelling of the polymer. But by the time your system will reach into the colon, and polysaccharide enzyme will degrade this gum and polysaccharide, or you will get the drug. Drug will be released in the colon. This is what we prepare, calcium pectinate microspheres containing the drug, anti-cancer drug. So here we use pectin solution 3% in phosphate buffer saline, right? Dispersed in isooctane having span 80, in which drug was dispersed well, and it is the pectin solution was containing 1% tween 80 as a surfactant. And this mixture is stirred at 1500 rpm for 10 minutes. We'll get the water and oil emulsion. Stir further for 10 minutes, we'll get the dispersion, and this dispersion is poured dropwise into the calcium chloride solution, 35%. We'll get the calcium pectinate microspheres. Because of reaction of the calcium ion with the pectin, we'll get the calcium pectin, which is water insoluble. So this is the same photograph of the calcium pectin microsphere without drug. This is with drug. And then just to ascertain the potentiality of the calcium pectin microspheres for colon effective colon delivery of the drug, the in vitro drug release studies were performed, and at different pH 1.2, pH 4.5, pH 7.5, and pH 7 of colonic fluid, which is containing some colonic content. So then we observe that maximum amount of the drug is released after 8 hours when your system will come contact with the colonic fluid. And then in vivo studies were also performed by administering oral administration of calcium pectin microspheres, and we observed that the maximum amount of the drug is observed in the colon after the 8 hours. Then Gour gum can also be used for developing a colon-specific drug delivery system. It is a hydrophilic polymer, natural polymer, swell in the cold water forming viscous colloidal dispersion. Because of gelling property, it retards release of the drug from the dosage form, as well as it is susceptible to degradation in the colonic environment by the polysaccharide enzyme. So we prepare cross-linked Gour gum microspheres. The 5% aqueous solution of Gour gum was poured dropwise into the castor oil having the span 80 and anti-foaming agent at 60 degrees centigrade. Stir for 10 minutes, then dropwise. Butylaldehyde was added, which will act as a cross-linking agent. So we'll get the hard microspheres which were dispersed in the castor oil. Then we centrifuge and wash it with the heptane and hexane solutions. We'll get the microspheres of the Gour gum. This is microspheres cross-link Gour gum microspheres without drug, and this is lower one is the with the drug. Then again, various in vitro studies were performed just to ascertain whether the sufficient amount of the drug is released in the colon or not, or where the drug is releasing. Then we observed that drug release in simultaneous colonic fluid we compare it, the drug release in simulated colonic fluid with or without red cecal content. Red cecal content means the simulated colonic fluid which is having the rich cecal content means it will be having the polysaccharide enzyme, right? So it is observed that when in vitro studies performed in the simulated colonic fluid without red cecal content, only 31% drug is released. But when we increase cecal content 2 to 4%, the release is increased from 31 to 47 and 61%. Then next approach is redox drug delivery system. As I told you, in case of inflammatory bowel disease, reactive oxygen species level is abnormally increased. I mean, 10 to 100-fold increase in mucosal ROCs observed in case of the ulcerative colitis. So Wilson et al. Synthesized a polymer, thioketal polymer, which is ROS sensitive, and they prepare a delivery system of this thioketal polymer for the delivery of siRNA. It degrades selectively in response to ROS in case of the ulcerative colitis or inflammatory bowel disease. Then active targeting, the dependent nano delivery system. It provides selected drug accumulation at the disease site within the colon using the ligands. Ligands will be acting as a vector which will carry drug delivery system to the disease organs. It improves therapeutic efficacy, reduces adverse effect interaction with the targeting ligand with the specific receptor overexpress on the colonic disease site will be used, and targeting moiety generally use transferring folic acid monoclonal antibodies in case of colorectal cancers. So here the nanoparticles surface is modified with this ligand like transferring folic acid or monoclonal antibodies, and this nanoparticle, ligand coupled nanoparticle, is filled in the hard gelatin capsule which is coated with the some pH sensitive polymer or some polysaccharides. So that will be or azoaromatic polymer, which are colon specific polymer. And this polymer will be intake in this capsule will be intake in the stomach and small intestine. When it will reach into the colon, the coating will be dissolved. The nano ligand coupled nanoparticles will come out from the capsules, and since these are nanoparticles are having the ligand, which is it will be taken by the receptor specific ligand, so it will be taken by the receptor which are overexpressed on the colorectal cancer, and we can get effective colorectal cancer targeting of the drug, anti-cancer drug. We can leave this, yes. So how we prepare this preparation of ligand encoded liposomes by using the carbamide chemistry. We prepare the folic acid coupled liposomes. The surface of the liposomes is coupled with the folic acid using the carbamide chemistry, and this is the folic acid coupled liposome containing 5-fluorouracil, and this is the this is uncoupled liposomes. This is the coupled liposomes, loaded coupled liposomes containing the 5-fluorouracils. So this is coupled, this is uncoupled. TAM photograph. Then these liposomes, the surface modified liposomes, are encapsulated into alginate bead. This is the procedure by which we entrap surface modified liposomes containing anti-cancer drug to the alginate beads. So liposomes to be encapsulated which are containing the drug were pelleted by centrifugation. Centrifuge it, the pellets will be taken out, be suspended in the distilled water, then mixed with the stock sodium alginate solution. it will be mixed properly with the sodium alginate solution. And this solution is dropped at the rate of 10 ml per minute through a syringe nozzle 22 gauge into the 50 ml calcium chloride solution. So this calcium chloride will act as a cross-linking agent. So then further stir for 1 hour to cure the calcium alginate gel beads. We'll get the calcium alginate beads, which are insoluble in the water. Wash it with deionized water, dry to a constant weight in a vacuum decycler. So two types of the beads we prepare. One is containing oxyplatin and another was containing this 5-fluorouracil. So this is the same scanning electron microscopy of the uncoated beads, encapsulated liposomes being 5-fluorouracil or oxyplatin. Then these beads are further coated. So all the possibility of releasing drug in the upper part of the GIT is prevented. Okay, so they were further coated with the intercoated solution, right, which was prepared by first making milky latex of hydrogen S100 using 1 molar ammonia, that is 1.5%, and stir for 1 hour. And it is containing triethyl citrate, 20% was added, and stirring was continuing for next 1 hour. And this intercoated solution dispersion was filtered by passing through 0.1 millimeter sieve, and this solution was sprayed with the help of nozzle on the bead. So we'll get the coated beads, right? So this is the same photograph of the coated beads, encapsulated liposomes containing 5-fluorouracil. And this is the same photograph of the coated beads encapsulated liposomes containing L-oxyplatin. This is in general the approach which we use, liposome-based colorectal cancer targeting. Liposomes bearing drug, right? First we prepare the liposomes bearing the drug. Surface is modified by conjugating the folic acid, right? And then these liposomes is entrapped in the calcium alginate beads, and this beads is further coated with the hydrogen S100. And this system, when you administer through the oral route, it will be intake in the stomach and the ileocecal region. When it will reach into the ileocecal region, the intercoating will dissolve, right? And uncoated calcium alginate beads will reach into the colon, where polysaccharide enzyme degrade this calcium alginate beads. The liposomes will come out, ligand coupled liposomes will come out. Since the surface of the liposomes is modified with the folic acid, so it will be taken up by the folate receptor over the colorectal cancer. So effective targeting to the colorectal cancer can be achieved. Similarly, we can prepare the multi-particulate system. Multi-particulate system is sometimes what happens, PSSD polymer releases drug in the ileum, which is then distributed in the colon. Because of pathophysiological conditions, there are change in the pH, and there are chances of releasing drug in the upper part of the GIT. So that instead of using the one approach, we can use multiple approach to rectify this problem, right? So multi-particulate system were prepared using same polysaccharide microspheres bearing the drug. Microspheres were prepared, coated with the polysaccharides, coated with the PSSD polymer that will be integrated in the stomach. The coating will dissolve when the pH will reach around 7 in the ileocecal region. And then polysaccharide microspheres, uncoated microspheres will reach into the colon. Polysaccharide enzyme will degrade this microspheres and drug will be released. So here the cross-link chitosan microspheres we prepared. Cross-link chitosan solution 4% in 5 W by V in 5% aqueous acetic acid was prepared. Drug was dispersed well, dispersed in the liquid paraffin having the SPAN 80 antifoam agent, stirred well, glutathione was added, toluene saturated glutathione was added, and cross-link this chitosan microspheres. Then solid microspheres were centrifuged, washed with hexane, dry in vacuum desiccator. Then these microspheres were cross-link microspheres were suspended in the organic solvent containing entry coating, means the cross-link chitosan microspheres were coated with the uracil containing. So these chitosan microspheres were suspended in the organic solvent containing the enteric polymer, emulsify. Then this will be emulsified in the liquid paraffin containing SPAN, tea, and foam. Continuously stir for 4 hours. Range it with an hexane dry in vacuum desiccated for 48 hours. Will get the coated chitosan microsphere coated with the uracil microspheres. This is the cross-linked chitosan microsphere without drug. This is with drug. Same photograph of the microspheres. Then again, various studies were performed to ascertain that how much drug is released in the colon when it will reach to the colon. So in the presence of without cecal content and cecal content, we'll see in the we'll see the drug release, right? And we found very less amount of that drug is released when it is no cecal content is no cecal content is present in the simultaneous colonic fluid. But when we add the cecal content in different concentration, the drug release is enhanced, right? Say here the 74% drug is released and 63% drug release is observed in case of the when coating is core coating ratio is having 1 to 10, right? Here is 74% drug release is observed when core coating is having 1:5. Then the cecal content which we use for this in vitro drug release study, we induce the enzyme. Sometimes it is not sure. We are not sure that whether this cecal content is containing the desired enzyme or not. So what we did, we induced the enzyme in the animal, in the colon, by feeding the this chitosan for 5 days. So for digestion of the chitosan, the bacteria will release polysaccharide enzyme. So it will contain more amount of the more concentration of the enzyme. So the cecal content will have the enzyme, large quantity of the enzymes. And so naturally by inducing the enzyme, the release of the drug is released 20 to 29%. Enhancement is observed. The next, I will take only 5 more minutes. So this is a very new delivery system we developed for colon delivery of the mesalazine. And this, we prepare nanocomposites. Nanocomposites of montmorillonite. This is earth matter. And this chitosan has been intercalated via ionic exchange by negatively charged montmorillonite, right? And this chitosan montmorillonite nanocomposite will get in which we incorporated mesalazine and trap in the mesalazine. So this chitosan montmorillonite nanocomposite remarkably reduce the ulcer index in the ulcerative colitis. It reduces ulcer index to 3.8. Ulcer index we observe 3.8 by this nanocomposite. If you compare with the chitosan plain nanoparticles, it was 7, and when you compare with the mesalazine plain, it was 9.85. So the tremendous reduce in the ulcerative index was observed because mentomolarity, apart from the say Mesilazine also acting as an anti-inflammatory drug, but mentmorolite shows anti-inflammatory activity also by absorbing the luminal antigens, increase of colonic mucus level, and possibly a direct modulating effect of the cytokine production by mucosal cells. So it absorbed this luminal antigens, which were increased in ulcerative colitis. It is well documented that epithelial barrier defect induced by the pro-inflammatory cytokines has been restored by the montmorillites. That is why we use this montmorillites for preparing this nanocomposite along with the cytosine. Dendrimers can also be used. As you know that dendrimers is in the skeleton of the dendrimers, whether it is first generation, 2 generation, or 4, 5th generations, there in the skeleton number of amino groups are present, and that amino groups can be used for conjugating that drug with the Azova bond, and we can make dendrimer colon specific dendrimer for colon specific delivery of the drug. Last one is the time release system, which is Pulsin Chem. It was first formulation introduced on the principle of time release for colon specific delivery, that is known as Pulsin Cap. It is having the hard gelatin capsule containing the drug. This is coated with water insoluble. Make it water insoluble by treatment with the formaldehyde, and this the mouth of this capsule is capped closed by the hydrosol plug, and then further it is closed by the cap of the hard gelatin capsule, which is entry-coated. And how it will act? This is Pulsin cap, which will when it will be administered through the oral route, it will reach into the stomach, it will be ineffective. But when it will reach into the small intestine, the intercoating of the capsule cap is dissolved, right? And this water soluble cap also dissolve, and hydrogel plug. This hydrogel plug will expose to the small intestine fluid of water, right? It will, when it will come in contact with the water of the small intestine, it will start swelling. So length of this will be increasing, and slow swelling of hydrogel leads a lag time that is predetermined. So by changing the length, we can increase the its residence time. We can increase transit time for releasing this cap, for opening this cap. So slow release of hydrogel lead to a lag time that is predetermined by length of the hydrogel plug. And this when it will be at the last end of the small intestine or in the colon, because of high swelling of this plug, the plug will be expelled out, the capsule will be opened, the drug will come out. So how, by this Pulsin Chem, how will we can achieve the drug targeting to the colon? So this is all about, this is the picture of the ceremony, award ceremony at Rashtrapati Bhavan, which where I received this Visitors Award 2018 for best research in biological sciences on 2nd May 2018 at Rashtrapati Bhavan. So this is one message I want to deliver to our colleague. We Indian believe in, let us not follow a path set by the others. Let us set a path for others to follow. Thank you very much.
Thank you. Participants may ask question if they are willing to ask. Thank you, sir, for sharing your very proficiency with us.
I would like to say something to our participants.
Yes, sir.
हमारे गुरुजी क्या बोला करते थे कि if there is a no question, it means your lecture is very good. or it is very well received by the patient, by the participants, and and there is a one aspect. Another aspect है कि आपका lecture इतना खराब था कि ऊपर से निकल गया. So from the participant, I would like to know which one is first option or second option.
I think option first, sir.
You are organizer, you will always say first, but
No.
recipients are different, no?
Yeah, but I think option first is correct for your question.
Okay, thank you very much
Thank
to
you, sir.
all and organizer and participant for patients listening of my lecture, right? And this topic I selected because I received a President Award on this concept. That is why I decided to deliver a lecture on this. And this will be a message, nothing is impossible, anything can be possible. मन से दिल से काम करना चाहिए बस, सक्सेस आपको हमेशा मिलेगी.
जी सर.
हमारे मेंटर हैं, वो कहते थे कि प्यारो फेंको तो कौन कहता है आसमान में छेद नहीं हो सकता है, फेंको तो एक पत्थर यारो दिल से. तो everything is possible. Okay, God bless you all. Take care.
Thank you, sir.
So anyway, if they are hesitating to ask me question in open platform, they can ask me anytime. You can share my email ID and you can share my mobile number also. They can ask me any question anytime, right? I will be happy to answer them. Thank you very much. Now, can I leave?
Yes, sir.
Thank you very much.
Thank you, sir. Our tomorrow's session will start at sharp 10 a.m. Thank you very much.